Thank you very much, Chris, for agreeing to give a Build a Cell seminar, and you can get started.
So thanks for inviting me and giving me the opportunity to present our work. So as requested, I have a couple of introductory slides on my background over deep time. So in 2005, I got a PhD in high energy physics. I hadn't taken any biology classes at all. And I would never have predicted I'd be in a biology department now. So I worked at Fermilab near Chicago, and then I also worked at CERN. Then I saw the light and moved into life sciences, working at the Sanger Institute, then later at Imperial College. And then in 2012, I moved to UCL and to start my own group in systems and synthetic biology. And this, so I've been there 10 years, over 10 years now. Our broad goals are to work on mutational processes, biological computation, and also microbiome engineering. And it's microbiome engineering that I'll be talking about today. And generally speaking, we were quite a mixed group. So we have biologists, physicists, mathematicians, and engineers. So it's quite a fun place, I think. So I'll start with the talk now. If anybody has any questions, feel free to raise your hand. I won't be able to monitor the chat though, but feel free to interrupt me if you like. So let's a broad introduction to this topic is in 2007, the Human Microbiome Project was founded. And although we knew about microbes within us, for hundreds of years, I think the Human Microbiome Project was the first time to map it completely. And it was showed that, we have microbes in every available niche in the body. But what I'm particularly interested in the work of my group is really the gut microbiota. This is where the biggest load of microbial cells is. And you can see this moving from the stomach, where it is low pH. Small intestine, there's an increase in bacterial load, but large intestine carries a huge number of bacterial cells, and this is a really complex environment. And two factoids that people always go on about: human body contains more microbial cells than human cells, and the microbiome contains hundreds more protein coding genes than human genome. So why is this interesting? Well, recently we've come to realize that we require this interaction for our health. So on the left-hand side here, you can see a cartoon depicting a healthy gut microbiome. So we have the intestinal cells here and the microbes here, and there's a homeostatic interaction between the two. And in fact, we now know that your immune system depends on presence of bacteria in order to be trained properly as as an infant, developing infant. And so what happens when this homeostatic situation breaks down? It's termed dysbiosis, which is a very vague concept, but essentially it just means the breakdown of the homeostasis, and there's a positive feedback loop whereby the immune system is upregulated, causing inflammation, which then gives rise to more colonization of more pathogenic bacteria, and and it's a positive feedback loop. And this has been shown to be related to many different disorders, ranging from diabetes, obesity, metabolic syndrome, and also the gut-brain axis, and cancer. And I say related to, we don't really know cause and effect, and it's quite difficult to tell. So another interesting thing that's coming to light recently, and I'll say a bit more about this later, is drug microbiota interactions. So it's clear now that the microbiota break down drugs, and they can also activate drugs. So, you know, there are 3 examples here. So E. coli converts this drug into its active form, and then these 2 bacterial species, so sorry, this one inactivates the drug, and this is a Parkinson's drug, levodopa, is converted to its active form by E. faecalis. So this is a huge area and will be really important in personalized medicine, and manipulating these bugs, particularly the ones that break down drugs, will be really important in the future. So how do we assay the microbiome? Well, we generally use sequencing methods, metagenomics, so either amplicon sequencing like 16S or shotgun approaches. And you know, this is applied widely. It's tricky data to work with, really, and can tell us certain things about the microbiome, the host microbiome system, but not everything. Here's an example of my gut microbiome sequenced twice. I don't know if I'm oversharing here, but you can see on one time I did it, I had a fairly a relatively healthy microbiota, so a balance of firmicutes and bacteroides. In this case, I have a massive prevalence of firmicutes, which meant I probably ate some sort of a burger or something before. So you can see that these readouts are not very quantitative, and we need new tools, I think, really to interrogate the microbiota, the host microbiota system. So that brings me to the concept of microbiome engineering. So one definition that I adapted was leveraging fundamental scientific and engineering principles and quantitative design to create or modulate host microbiome systems. And there are various different approaches to engineer the microbiome. And I'm not, this is a general definition that could apply across the human microbiota, the gut microbiota, the skin microbiota, the oral microbiota, roots, soil microbiomes, and etc. But as I say, I'm mostly focused on human health. And there are various different ways you can perturb the microbiome. So diet, obviously, probiotic, and then FMT, fecal microbiota transplants, microbial consortia, phages, and engineered live biotherapeutic products. And these are all the various different targets. That was just a representation. So just to emphasize this point, I think microbiome engineering is not just about designing new microbe systems or treating some malady. It's also about understanding. So actually understanding the system requires careful perturbations and measurements, and I think that's important. It's not just about achieving health benefits. We really need these tools to understand this complex system. So some terminology. So you sometimes hear probiotics, sometimes hear live biotherapeutic products. So the technical term for medical applications is live biotherapeutic products, and this is from the World Health Organization. And it shows the main difference between probiotics is this is a food supplement, and it is not regulated in the same way as a medical product. And that's where live biotherapeutic products come in. So this is what we generally use LBPs to refer to these microbes or microbial formulations. So live biotherapeutic products is an active area in the in industry. So a lot of interest has been around fecal microbiota transplants. These are highly efficacious for C. difficile infection, and there are lots of studies out there for applying it to different areas. But also people are developing microbial consortia and single strains to target certain problematic disorders such as IBS, for example. What we're talking about today is engineered live biotherapeutic products. So this is really genetically modified bacteria used within people. And there were lots of current human trials with engineered live biotherapeutics, most of them in the US. You can see that they range from a lot on targeting cancer, but there are others as well. Many use lactic acid bacteria because they were traditionally probiotic strains, but also E. coli. So if you're interested in this, and this surprises quite a lot of people that this is already in clinical trials, you can check out this review by Jack in our group. So we've been working on this in this area for a few years now. We do microbial systems modeling, so that's trying to model the data that we get out of microbiome studies, try and make inferences about interactions in the microbiome. We've worked on synthetic microbial communities. So this is where we engineer microbial communities in order to understand how they function and to manipulate them. Biosensors, engineered probiotics or live biotherapeutic products, and that's what I'll talk about today. We've also worked on some new novel host microbe systems, C. elegans, which was fun. So now I'm going to focus mostly on cancer because that's what we've been interested in, and I'm sure a lot of you are aware of this area. But there's a lot of activity on microbial therapies for cancer. So, in the 1890s, there's this guy William Coley, who's now known as the grandfather of immunotherapy, and he discovered somehow that injecting streptococcus directly into the tumor of a patient, an inoperable tumor, almost certainly fatal, it could cure the patient for a number of years. And this was essentially the triggering the immune response to attack the cancer through these bacteria. More recently, people have engineered. So we know that bacteria will inhabit a tumor because it is a hypoxic environment and it's out of the immune. Generally, there's no immune response there. So bacteria will colonize tumors naturally. And people have been engineering the bacteria that colonize in order to deliver chemotherapeutics. And Saldonino's done some very cool stuff at Columbia, engineering an oscillator within these cells to deliver. So this was in E. coli cells to deliver a therapeutic into a tumor in vivo. So this is a big active area. So these approaches generally deliver chemotherapeutics. So you're trying to target directly the cancer. But actually, there's a growing number of studies showing that that the bugs that colonize the tumors can have can give rise to poor prognosis in patients. So F. nucleatum is kind of the first one that's been really characterized. This is a an oral commensal. But if it's in the tumor of patients, they have worse outcome. And there's a number of mechanisms, depending on signaling, but also delivering toxins as well. And this can be causal. So this can actually cause colorectal cancer. And then you have other interactions like E. faecalis, E. coli, and B. fragilis that can deliver different toxins to the cells and be cancerous. So we think that targeting these bugs, rather than the cancer cells themselves, could be beneficial for adjuvant therapies. And this is further reinforced here because it's a very important. It's become a very interesting area in the terms of cancer immunotherapies. So how patients respond to immunotherapies will depend on the bacteria that are living within their gut microbiome, but also within the tumors that they have. And I'm not going to go through all these mechanisms, but there are various different bacteria that are known to interfere with drugs, these immunotherapy drugs. This was an interesting paper just either this year or last year. And what they showed in this paper was they showed a number of things, but one of the nice things they did is they took cancer patients and they looked at the survival of cancer patients who'd previously taken antibiotics that target anaerobic bacteria. So the anaerobic aspect is a proxy for tumor colonizing bacteria. And you could see that if they'd received antibiotics, they had a better outcome because these bugs that are in the tumors have been targeted, and they actually developed an antimicrobial using liposomes to deliver drugs. And this is really cool, and it's kind of targeted to low pH regions, but it's still quite broad spectrum. You're targeting all the anaerobes, and this is this can be problematic for cancer patients if you're going to give them antibiotics. You don't want to give rise to further problems that are complications that are related to the delivery of those antibiotics. So what we've been doing the past few years is looking at using bacteriocins, and these are small antimicrobial peptides which are naturally produced by most bacteria, and they're there to target similar species in some cases that are in the same niche. So they're like a niche creation tool, and they can be very specific to certain species of bacteria. They can also be broad spectrum as well, but they're generally recognized as safe. So the most famous one is niacin, which is used in food. I don't know if this is just European E234, the E number system, or it's international. But you can, if you can look in your food, and you'll find E234, that's niacin. And what's interesting is they can be engineered or modified. You can create different chimeras with different modules, and that's what this paper here from Ron Weiss's group did as well. So we've been using them. Now, why, if these are so good, why are we already not using them in treatments, you know? And people are working on using them because they're important for antimicrobial resistance. But the challenges, the main challenges, are that antimicrobial peptides are sensitive to degradation, and therefore administering them effectively to the target site can be challenging. And here's one example from a study that uses this Galleria model, which is like a moth grub larva, and they had 5 different bacteriocins that were active in vitro, but 4 of them were not active in this Galleria model, and that's because they're getting broken down. And you can see here that none of them were as good as the antibiotic that was given as a control. So that's the main challenge. So what we've been working on is a way to get around this, is combining this this property of bacteria to colonize tumors automatically, and then engineering those to target the microbes in there. So here's E. faecalis growing in a tumor, and then here's the E. coli Nissl that we generally use, secreting the bacteriocin to target E. faecalis within the tumor. So this has the potential to be highly targeted and effective at removing, you know, very specific species of bacteria. So we're not the first to attempt this. There's been a number of people who have engineered different. This is generally E. coli, engineered E. coli to target pathogens by expressing bacteriocins. So all of these are in the context of antimicrobial resistance. It's not cancer treatment. But what you can see here is the different. So this is from a paper, nicely summarized the previous work. So people have tried to target Salmonella, VRE, Pseudomonas, Salmonella, and drug-resistant E. coli. But you know, what we've done is kind of tried to make take a really generic approach, and you can see the preprint here. We've been working with a company, Singulon. So Singulon have a library of bacteriocins that we can screen against any particular pathogen, and then the idea is to use symbio engineering biology tools to make a generic delivery platform. This is the best I could get out of DALI of engineering biology. I won't be putting it in any papers, though, as recently has been done a few times. So essentially what our platform does is it just uses the Moclo expression platform, and we've modularized the parts inside the expression system. So we have bacteriocin coding sequence here, we have secretion signal, and then the standard parts, RBS, terminators, etc. So we can modulate the secretion tags for how the bacteriocins exit the cell, and we can change the plasmid copy numbers as well. So here's a depiction of the peptide being secreted and targeting the species of interest. So for this study, we decided to look at E. faecalis and E. faecium. As I said, E. faecalis is already linked to cancer development. E. faecium, I mean, they're both of interest in terms of antimicrobial resistance as well. But they also seem like a good model organism to demonstrate this platform on. So we also started out with two bacteriocins, so Nt and Nt-B, and these are both produced by Enterococcus strains. And the secretion system we're using is the SEC system, which transports across the inner membrane, and then generic transport occurs across the outer membrane. And we can modulate these signal peptides, and I think nobody's really looked at that, the effect of the signal peptides on secretion in a systematic way, and you know, we looked at these as well. We can incorporate those into the system. So here's kind of the standard approach to verify how your engineered strains kill the target. So we have a lawn of E. faecalis, and we grow up the cells that the engineered E. coli, it's E. coli Nissl. We grow them up and look at these zones of inhibition. And what you can see here in each column is the different signal peptides. So this tells you how much is being secreted, and the two different bacteriocins. So we seem to get more killing with Nt than Nt-B, which is what we see in the literature as well. And you know, we have different secretion signals that work the best with different bacteriocins. So there's some interesting design, machine learning problems or you know, problems that can be solved with these a complex design space, I think is the right way to put it. So we want to, so this is fine, right? This is what everybody does if you study antimicrobials. You get a minimum inhibitory concentration. But when you're designing engineered live biotherapeutic products, it's no good loading the cells up with bactericin producing systems and reducing their growth rate. So we want a balance between growth rate and killing efficacy, because you know, they're inversely related. So we spent a lot of time looking at co-culture dynamics. So the idea here is we grow the target strain up with a control. So this is just E. coli Nissl, that doesn't express any bacteriocin, and E. faecalis or E. faecium outgrows the control, the E. coli in this medium. And then we do the experiment with the engineered strain, and what we want to see is the target strain driven to zero. In order to do this, we use this tool Flopper that was developed in my group by Alex Federek, who's now at UCL as well. He's got his own group. So this uses calibration of the flow cytometer or plate reader. We're just using plate reader here. So it allows us to do the co-culture experiments with only labeling the E. coli. So we don't need to label the target strain, which is really quite useful. It means we can extract co-culture data from only labeling E. coli with fluorescent protein. So, and these are the kind of plots that we're looking at here. So this is kind of the equivalent of the inhibition zones. So each one of these, so at the top here we have NT, at the bottom we have NB, and these are with the different signal peptides. So here is the control. You can see that this is all E for careless. E for careless outgrows E. coli, which is this one. Then when we put in the engineered strain producing the bactericin, E. for careless growth is driven right down. Some cases we get it down to 0. And this is the equivalent in MB, and you can see that we get less killing with NB than NA. So we can also look at the dynamics as the seeding changes. So if you look across here, you can see, let's have a look, yeah. So if you look across here, you can see this is an increase, a decrease. This is the ratio is 9:1, but at the bottom is the seeding. So it's seeding across here and the initial ratio here. So the total amount versus the ratio, and you can see you get different behaviors. So here's E. for careless outgrowing the E. coli. And then you get the opposite here. You can see that E. for E. coli will eventually outgrow. So depending on how you seed, you get different results. What we want to see here. So this is the one that we see at the start. Oh, this is the main one we started with in the middle. What we want to see is that the E. coli still outcompetes E. for careless even at low seeding ratios. So this shows how the liquid culture and the inhibition zones are correlated. So that's interesting. It shows that what we're measuring in the solid culture is correlated with what we measured in the liquid culture. So because we've set up this the SynBio platform, we can then start combining bactericin. So we included different versions of NT and NB combined, and we can show that we get much more killing when we combine the two bactericins, and that's shown here. They're the solid culture and the co-culture. So now we're really driving the E. for careless to 0, and really out competing it. And here you can see that the combinations in solid bars pretty much always does better than the individuals. So here's the individuals for one signal peptide. Maybe this one doesn't do as better, but mostly combining the bactericins does better than the individual ones, as you might expect. We also develop mathematical models for this. So we use like a Lotka-Volterra pairwise model to model these co-cultures. So here you can see one set of data. So there's the monocultures, here's the co-cultures, and we fit a Lotka-Volterra type model. And what we can do is estimate the growth rates, but we can also look at the different, I call this killing curves, but it's basically the response of the E. for careless to the E. coli bacteriocin production. So you can see you can measure these different properties of the bacteriocins indirectly through doing this Bayesian statistic analysis. So a lot of people ask about resistance or susceptibility, and we've done passaging experiments, and we can you can see resistance emerging eventually. So by passage 4, so if you look at this, this is what you've been seeing earlier. This is E. faecalis growth here, and after 4 passages, 12 hours each, you can see that it eventually does start to grow back, okay, and outcompete the E. coli. So the idea here is that we will add, so with 3 bacteriocins, we should be able to defeat this, or even we could even try 4 bacteriocins. You can see that this effect is not as big in the combined NTB case. So everything I showed was for E. faecium, but you can also see the similar killing for everything I showed was for E. faecalis, but we can also see the same killing for E. faecium. In some respects, it's slightly easier to kill than E. faecalis. So we've actually got other bacteriocins that target this one as well. Because we've got this platform, we can do library generation. So this is one of the advantages. So this is really cool. So we took the NTA system and built a library of ribosome binding sites and promoters. That's depicted here. And we can just build the library and then screen on plates by killing zones. What's interesting is that just by doing this simple proof of principle, we were able to improve on our rationally designed system. That's in pink. That we had two or three systems that perform better by just selecting the RBS and promoter combinations, and the 2 ones, D and E, you can see use these parts. So we think this is this is going to be really useful for developing and optimizing the targeting of different bugs. Additionally, we can also use other synthetic biology parts. I know it's kind of obvious, but this really opens up what we can do. So here we combined with the p-arabad promoter and built an arabinose inducible bacteriocin production. So what you can see here is the arabinose. So this is the response of kind of the biosensor part, and then this is the inducible, the characterization showing the killing zone as we increase arabinose expression concentration. Sorry. Okay. So we're also targeting, so everything I showed was for E. faecalis and E. faecium, but we're developing this platform to target F. nucleatum and B. fragilis as well. So we've already done some screening. So Kim in my group has screened various different bacteriocins using this library from Singulon against F. nucleatum and B. fragilis, and we have at least 4 bacteriocins targeting F. nucleatum and B. fragilis that we want to incorporate into the system. just quickly, we've got, just want to say that we in parallel, we're always kind of trying to engage and think about societal issues. So we've also developed a survey to gauge public's interest or knowledge of microbiome engineering. We got 100 full responses on the first run, but the second run we're setting up now. I think I'll just jump to this part, which was so we're looking at cancer patients. So you might bear that in mind. And in a way, that's why we ask cancer patients. So we asked them, would they be comfortable with taking engineered live biotherapeutic products? And, most of them, or a lot, perhaps surprisingly or unsurprisingly, most people, if you've got cancer, will be prepared to take anything to fight the cancer. But we were pleased by these results and other results that we got. That people, I guess as opposed to like GM food, there's a good return for the risk in terms of a cancer treatment. Whereas for GM food, it isn't, perhaps the return is not worth the risk. So this is interesting to see and to get responses from the public. So that I'll end there, and I'll just say we thanks for listening, and we constructed this generic antimicrobial delivery device, and we think it's useful for lots of applications. I think we're focusing mostly on this cancer application, but it's much wider potential. And we focused on Enterococcus species, but we're moving now to the actual interesting cancer-causing agents. So I think there's definitely scope for designing these better. And I think we can use these tools to understand also the secretion systems. I think we need to understand these a bit more. And obviously we need to verify in ex vivo and in vivo models. If you're interested, the preprint is here. So I'll just say thanks for listening, and most of the work here was done by Jack and Linda, Kim, Chania, and thanks to my collaborators and funders. Thanks.
Thank you so much. That was a great talk. I really learned a lot. I guess I'm not up on that literature as much. There are questions in chat. Can you see chat?
Yeah.
If you could read a question out loud for the recording before answering,
okay.
that would be great.
So the first one is, in your co-culture experiments, are the changes only due to the competing bacteria species? Are there other factors, like could you change co-culture ratios with small molecules or other factors? So yeah, so we use the same media, and in those experiments we just changed the seeding ratio and the total number and the total OD. So that's what's changing there. We haven't looked at other changes such as temperature or small molecules as you suggest there. So we haven't looked at that, but it's interesting. It's an interesting idea. Second question, out of curiosity, what's the biggest public concern in regards to microbiome? Do you think there's a difference between the public perception in the US versus in Europe with the anti-GMO fear-mongering on your side of the pond? Yeah, I mean, yeah, it's a very good point. I joke that one of the only benefit of Brexit was that that we're no longer necessarily bound by Europe's fear of GMO. But yeah, it's an interesting point, and I think This is exactly my point. Is like if we're talking about GM food, I don't think perception will change in Europe anytime soon. But if we're offering life-saving treatments using GMOs, this is how we have to shift the public attitude. I think it is this risk return or benefit cost analysis. But yeah, it's an interesting question. So the next question, could you speculate if those antimicrobial delivery technologies could be engineered into a synthetic liposome, not live engineered bacteria? Yeah, so I think people have expressed colicins from liposome artificial cells. So it is possible to do that. People have already done it, and yeah, it's something that someone at my collaborator and colleague at UCL, Michael Booth, is working on at the moment, actually. So yeah, it's definitely possible. So here's another question: is the gut microbiome a bacterial consortium where they communicate and share resources across species, or it's more like a co-culture where everybody grows on their own? Sorry if it's a naive question. I'm not an expert here. No, that's a good question, and even the experts would disagree to some extent. There's this whole thing about whether it's competition or cooperation in these communities, not just the microbiome, but more broadly. We tend to want to classify things in terms of one or the other, but it's probably a lot of both. But what people have shown is competitive interactions provide more stability than cooperative. But it's a bit it's controversial, and you'll get different papers coming out every now and again that said various different things. But it must be a mix of competition, which is what I'm calling growing on your own, versus cooperation. So it must be a mix. And plus you have also the same interactions, but with the immune system. So it's highly complex.
So I have a follow-up question if there are no more questions. You said something really interesting when you said competitive relationships are actually more stable than cooperative. That seems counterintuitive. Again, I'm not a microbiologist either, but that seems counterintuitive to me. Why is that?
Okay, I'll attempt to explain in one sentence, but basically perturbations propagate in cooperative networks easier than in competitive networks. So if you imagine one species goes down that everybody else relies on, then the whole thing crashes. So that's the idea behind this instability. So yeah, so that but I think you probably need you need both. I think. Oh, for sure they compete. For sure they cooperate as well. I mean, horizontal gene transfer of antimicrobial resistance genes is a form of cooperation across species. So for sure they're doing both at the same time.
Oh, yeah, that makes sense. All right, last call for questions. And if there are no more questions, I just want to say thank you so much again, Chris, for the talk.
All right. Thanks for inviting me. Yeah.
Thanks everyone for questions, and have a great rest of your Monday or all of your Monday, depending where are you.
Okay. Thanks, everyone.
Bye.
See ya.